I'm actually a clinician by training, and I got into sex differences more recently. And hopefully what I'll be able to do today is tell you a little bit of why I think it's relevant to this discussion. So I'm specifically looking at sex chromosome aneuploidies and why it might be a really interesting model for us to focus on and trying to unpack some of the things that we still don't know about sex differences work. That might be a little bit different from a lot of the other talks, which have looked more at the mechanisms as they relate to hormones. And part of what we'll cover is really understanding a little bit more about the phenotypes of sex chromosome aneuploidies, and as well as the genetics underneath that might provide a mechanistic framework for some of this discussion. So I'm sure everyone who's in the room who's been doing sex differences research is obviously very familiar with the seminal study by Phoenie looking at guinea pigs and really showing for the first time that there is a masculinization effect on the brain at a very early part in development in prenatal exposure to testosterone. And it's really fascinating to think, as a lot of the talks earlier today, and this has been refined and elaborated in Dr. McCarthy's talk, but how this can have enduring effects throughout the developmental stages and not just in the prenatal period. It's been discussed already what the organizational-activational hypothesis is, and really a lot of the emphasis, like I mentioned, has been on how this is mediated really through the hormonal effect. So while there is there's an indirect effect, it starts in the genetics, so really looking at the XX and the XY. And as was mentioned before, it's really the presence of the SRY gene that triggers all these downstream effects through testosterone and estrogen and prostaglandins. And what we want to do, hopefully today in today's talk, is bring a little bit more attention back to the XX and the XY and try to understand and see if there might be actually more direct effects than has been really realized in the recent literature. So a lot of the discussions today has focused on the animal research, and it's a lot easier to look at this, I think, because there's more evidence of sex-type behavior. So as Dr. McCarthy mentioned, we can look specifically at things like mounting behavior, right? You can look at the difference between males and females in terms of their sexual behavior. As titillating as it would be for a study section, I think it's a lot harder to get a grant funded to do this in human research, right? We can't really look at sexual behavior in humans and really say definitively, oh, there's much more male versus female sex typing in regards to that. But luckily for us, there has been a lot of work in the psychological domain, really trying to identify if there are genuine male-female differences across a number of different aspects. And also referenced earlier, although the effect sizes are much more modest, so like we had, like was earlier presented, it's usually below one standard deviation. So it's not nearly to what we're seeing in some of the sex type behavior in animals. But there are certain things. So for example, this is Melissa Hines' review. She looked at the topic several years ago. But things like core gender identity, right? Males, boys, and girls are going to strongly identify as either male or female. And, again, there's a social piece to that because probably a lot more youth nowadays don't really conform to a gender binary. So there's clearly some differences there, but it's very robust. Other things like sexual orientation, that's obviously very debated, but again, they're pretty robust genetic or sexual orientation identification according to your genetic sex. And then as you get into more social behaviors, it becomes a little more tenuous. You have childhood play that ranges between effect sizes of 1 to 2, playing with sex-specific toys, girls and boys, different kinds of playmate preferences. And then this is the area that we'll focus the talk on today, is looking at more biologically related aspects of psychology. So cognitive abilities, motor abilities. And again, this is where we start to see the differences between the sexes might be more modest or may not exist at all. So we're looking at Cohen's D here, of anywhere ranging from 0.3 to 0.6. So, in psychological research, we tend to consider this to be fairly moderate, but it becomes really debated how much signal there is there compared to noise, right? And obviously, human behavior is incredibly complex. I'm a psychiatrist, and to be able to say we can characterize these behaviors in a consistent way and break it down into quantitative units is really difficult. If you were keeping up with academia 10 years ago, some of the debates about cognition and sex differences in cognition, there was a lot of brouhaha when Larry Summers was the president at Harvard at the time. And he was talking about why women are underrepresented in STEM domains. And, you know, he brought up at a private talk, he was talking to at a conference, he mentioned several aspects. One of the things he said about underrepresentation of women is simply they weren't able to keep up with the output or the work demands of an 80-hour work week that men were able to, in this, both in science as well as other fields. But he also cited biological data suggesting that there are these real differences between the way men and women process science and technology. And, you know, obviously this caused a huge uproar, and it's really no coincidence that he got replaced a couple years later by the first female president at Harvard, Drew Gilpin-Faust. But it brings to mind then, how can we really see whether or not these cognitive differences are real, even before we can make some kind of estimation whether it's biological versus social? There's a group, Janet Hyde's group at University of Wisconsin and Marshall Lynn at Berkeley, who tried to approach this question by looking at just overall academic achievement or academic outcomes across males and females. And they did a meta-analysis, and they included like 200 some odd studies, which averages over about 1 million some odd students. And if you really look at the comparison between the groups, you can see that it's distributed in such a way that there's, in the meta-analysis, very little evidence that there's a difference in mathematics performance. So it converges at 0 between males and females. And again, when you look at the variance between these studies, they're pretty much around 1. They're very similar in the distribution. They looked also at longitudinal educational data. And they found very similarly, if you follow these kids over time in these educational tracking, that it was also very little differences that were significant between boys and girls. And one way to really think about this is, you know, is there really a social piece, is to think whether it's restricted somehow to what we're doing culturally, right? Like, is there something about American society that might say, okay, we have some more equivalence between males and females in mathematics performance? And if you look at the international data, this is pretty consistent that, again, that at least in mathematics and science performance, there's probably not a robust difference between inherently the way males and girls and boys are performing. So every 3 years, the OECD, the Organization of Economic Cooperation in Developing Countries, I believe, they do a triennial report really looking at 15-year-olds across a number of their member nations and collaborator nations. And they just assess how are the kids doing in terms of academic achievement. And you can see that although there's a number of differences across regions and across economies, that the circles here are the boys, the filled in dark circles, and then the diamonds are females, girls. Between 15-year-old boys and girls, mostly they're overlapping regardless of what culture you're in, right? And if there is a difference, the effect size is probably quite modest. So this is for mathematics performance. And this is the 2015 data, which is the one that the most recently published, but it's also the case for the 2012 data going back. If you look at the science performance across these different cohorts, you get a very similar pattern. And in some countries, you'll still see that either they're close or that there might be females outperforming males and whatnot. So if we look at it purely at a behavioral phenotype level, so we're looking into seeing, okay, in terms of adaptive functioning academically, is there going to be a male-female difference? Is there some kind of pertinent sex difference in performance? And I would say the bulk of the data really suggests that there isn't. So whether or not there are cognitive differences, at least in the typically developing population, I think there's still not a convincing amount of evidence. But here's something that never gets discussed that I do want to show you from the same report. This is reading performance data. across all of the same countries. And you can see all the girls are vastly outperforming males. It's not even close. And for whatever reason, we never talk about that. But, you know, I do want to say that it makes the picture a little bit more murky, right? Like, I think there's enough to say here. Potentially, it's worth really investigating more. Are there certain biological principles that, you know, similar to the animal research, might drive some of our understanding of how cognition works, and if there are true sex differences in cognition and learning? So as a clinician, this is a really relevant question. So I think partly, you know, it's a hotly debated topic in the lay media and pop psychology and whatnot, but there's true relevance in the clinical world. And that's because sex differences are profound, as mentioned before, in a lot of clinical diseases, so particularly in psychopathology. So in neuropsychiatric disorders, whether it's prevalence, the course of disease, and as brought up earlier, whether it even has to do with treatment, there's really you know, a lot of good evidence to show that males and females are differentially represented in all of these areas. And again, if you think about it within the organizational-activational framework, these are also time-locked. It happens in developmental stages. So the differences that you see really differ depending on the age of the kids that you're working with. If you look at the CDC data of just the incidence of psychiatric disorders, period, there are substantially in neurodevelopmental disorders. So these are the disorders that occur early in childhood. There's a substantial male predominance. Whether it's ADHD, learning disorders, ASD, it's anywhere between 2 to 1 to the disadvantage of boys compared to girls, all the way up to 5 to 1, 6 to 1. And then what's additionally interesting is if you now look at adolescence, so in the activational phase, so what would be the equivalent in the developmental stage, where you have, again, this surge of pubertal hormones shaping the brain again, those differences shift a little bit in a different direction. So mood and anxiety disorders, so major depressive disorder, generalized anxiety disorder, separation anxiety. While the neurodevelopmental disorders, the ones that present early in development by early childhood, things like ADHD and ASD, they retain this male predominance throughout life. Mood and anxiety disorders, which tend to be equivalent between boys and girls prior to puberty, so there's usually a 1-to-1 ratio, at least in the incidence, it emerges that it becomes female predominant around adolescent for whatever reason. And, you know, there's a lot of thinking about why that occurs. And one of the obvious things to think about is really the hormonal influence. And so, you know, when I'm in clinic, I'm running around between my clinics, and in my ADHD and ASD, the autism clinics, it's all these young boys that you end up seeing. And then you go over to the eating disorder units, and it's all adolescent females. And really, what's driving that, right? Like, so it's something that's just really obvious on an anecdotal level. But our understanding of that mechanistically is something that is still leaves much to be desired, and hopefully some of the science that's presented earlier is going to answer those questions for us so we can improve the outcomes for these kids. So as I was finishing my clinical fellowship, I had the fortune of thinking about these in a more systematic way. So, you know, like other people have referenced in the animal research, so not just in male and female rats, like, you know, all these people have been publishing on male rats only, but that's the same thing in human research. A lot of our clinical paradigms, our algorithms, are based on the fact that we've studied male populations primarily, and we're just extrapolating the same data, the same algorithms and the same treatment strategies to females with the assumption that whatever the biology is going to affect them in the same way. And like we're hearing, there's good evidence to suggest that that really may not be the case. So I ended up doing a research postdoc. I stayed on at Stanford in Alan Reese's lab, who's sitting here in the back of the room, and he's been doing work in neurogenetic syndromes for decades and included Turner syndrome, so sex chromosome aneuploidies. And that was the first project I got staffed on when I came into the lab. And sex chromosome aneuploidies means literally an abnormal number. So it's a count where in the X and Y, so we're diploid organisms with 23 sets of chromosomes. So while every other autosomes are paired equally between the paternally and the maternally inherited, between the sex chromosomes, the X and the Y, there's differences now in the number that you have. In a typically developing female, you would have two X's, a 46XX karyotype. And then in males, typically developing males, you'd have a 46XY karyotype. So what happens in the sex chromosome aneuploidies? There's one called Turner syndrome. There's only one X chromosome rather than two. It's a haploinsufficiency largely, right? It's an entire chromosome loss. And this is actually much more common than you would think. It happens in 1 in 2,000 live-born births. So remarkably, given the fact that it's so common, there's very little research in this area. The sex chromosome aneuploidy research community is actually quite small. In addition to having this genetic abnormality or related to it, it's the most common cause of primary hypogonadism in females, right? So in development, around the 15th week of pregnancy, their ovaries start to involute, and often, at least in the monosomic karyotype, they're born with streak ovaries at birth, or they're non-functional, essentially. And Turner syndrome is actually the only monosomy that exists in humans. So we'll talk about that a little bit later. There are probably some built-in mechanisms for that, but this is the only one that's compatible with life. With none of the other chromosomes can you be missing an entire copy and make it to full term. The opposite sex chromosome aneuploidy that we're looking at is something called Klinefelter syndrome. So this, again, is one of the most common genetic disorders in males. It's 1 in 600, and in this instance, rather than having one X chromosome, you have two in addition to the Y chromosome. And again, this is the most common cause of primary hypogonadism in males and adults. So in this instance, the low testosterone emerges as later, though. So while in Turner syndrome happens earlier in life, in Klinefelter males, it occurs at puberty. But it presents this lovely model now for us to start looking and segregating in humans this unique way. It's a unique paradigm of really trying to understand what are some genetic effects of sex differences and what are some hormonal effects. So you can see that there's essentially a linear dosage relationship when you talk about it at the whole sex chromosome level. 45X, the Turner syndrome girls, will have one X chromosome. The typically developing females have two X, two sex chromosomes, two of them X, and then in the males, XY, and then in Klinefelter, XXY. This is complicated a little bit about the differences in the hormonal status, which I was just referencing. So Turner syndrome girls will be deficient in both estrogen and testosterone early in development. And then you would see typical hormonal profiles in the males and females. And then in the Klinefelter boys, they start to have testosterone deficiencies, usually emerging in the around the prepubertal period, so just as they're entering puberty. So if we take a look at the phenotype, can we really distinguish these two groups? If we have now this linear model of gene dosage, of entire sex chromosome dosage. And interestingly, there does seem to be some patterns that would be consistent with that. So overall, both of these groups, both Turner syndrome and Klinefelter syndrome, they have normal intelligence, so it's in the average range, but probably it's 5 to 10 points lower than their siblings who are typically developing. And what's driving that is if you look at the subdomains, they have this split between the verbal IQ and perceptual or nonverbal IQ. And they trend in opposite directions. So in Turner syndrome, they tend to have preserved verbal IQ, but impaired perceptual or nonverbal IQ. And then in the Klinefelter group, they tend to have impaired verbal IQ while they have retained perceptual IQ. They both, however, have, in higher cognitive functions, so things like executive functioning, they have very similar problems. And then there's differences in things like arithmetic or verbal processing between the groups. And we went and we looked at this across, we just did a weighted mean analysis of a number of studies. So they've been publishing on this data for several decades, the psychological data. And it's really consistent. And what's interesting, you can see in Turner syndrome, this is the pattern. So, this is the 45X, the monosomy. And then you see the flip pattern in the Klinefelter group, at least in terms of the subdomain criteria. And this is true also for other trisomies. So I included the XYY group because there's a number of studies that also include the psychological data for that. And that seems to support that maybe there are these cognitive domains that might follow some kind of genetic dosage model. If you look at the clinical phenotype, not surprisingly, it follows along with what we're seeing in terms of their cognitive abilities. So in terms of executive dysfunctioning, which is prominent in both groups, you see much higher rates of ADHD in both. One of the junior faculty in our lab, Dr. Green, is looking at it more carefully. And there are some subtleties even within ADHD. So the phenotype of ADHD might resemble more of what you typically see in boys with ADHD than girls with ADHD, in the sense that they tend to have more of the hyperactive and impulsive dimension compared to what is typically seen in a lot of girls with ADHD, was the inattentive type. But also in terms of their learning and cognitive abilities. Dyscalculia, which is a math learning disability, you'll see that in up to 55 to 75% of girls with Turner syndrome, they would meet that criteria. In contrast, that same proportion of boys who have Klinefelter syndrome would meet criteria for a learning disability in language-based domains. So we were able to get some data from a colleague, Judith Ross, over in Philadelphia. And qualitatively, if you look at the analysis in terms of, these are structural analysis of gray matter volume, so just brain volume in different regions. You can also see that there's a nice homology between the regions that are affected, again, following this dosage model. So in Turner syndrome girls who have one less X chromosome, they tend to have all these differences that you would see both in the temporal insular area, where they have increased volume relative to typically developing females, and then decreased volume in the parietal, somatosensory regions, and also the occipital regions. And then the reverse is actually true in terms of gray matter volume. Klinefelter boys, relative to typically developing boys, have less volume in those same regions, increased volume in those same regions, and then less volume in the temporal insular regions. And when you look across all three, you really see that these stack up together, showing that it really does follow this linear pattern that is consistent with having 1, 2, or 3 chromosomes. So, it's been really argued, at least in neuroimaging, our field, whether sex differences are real, So one of the things that have been most consistently reported is that women and men have different global brain volumes. So there's been a real push to argue that a lot of these differences it's simply a scaling problem, right? We have an allometry issue. Because men have larger brains on average than women, you're going to estimate that these regional differences are just an artifact of that. But if you really think about it, and if you really controlled for that in our particular data, we found that that's not the case. Because in Klinefelter syndrome, the volume is actually less. It's actually the opposite of what you anticipate. And yet they still had the opposite of pattern of what you would see in the Turner girls, right? So it doesn't match. You can't explain it all away just by global brain volume size. So this is really a unique way that we found that these differences exist even in spite of global brain volume differences. So what is really interesting, just to kind of encapsulate this a little bit, what's really interesting about this, I want to bring our attention back to the fact that you really don't see testosterone deficiencies in Klinefelter syndrome until puberty. So all the data I just presented to you, we specifically looked at prepubertal kids. So what that means is the Klinefelter boys and typically developing boys are hormone status equivalent. They are not testosterone deficient in this early age. So this is a model that departs a little bit from the organizational-activational hypothesis. The prenatal hormonal milieu for Klinefelter boys is not, so the differences that we're observing putatively is not related to the fact that they have less testosterone early on in development. That might be the case starting from adolescence on to adulthood, but what I'm really presenting to you is some evidence that we think suggests that there's an alternative hypothesis for this. And that's why, obviously, we want to bring our attention back to the genetics. So because it follows this linear dosage pattern, and it happens in the absence of these testosterone differences, at least in the Klinefelter boys, that there might be some mechanisms that are occurring through a direct influence from the sex chromosome genes, rather than from a mediated through hormonal differences along the way. And, there's also additional animal evidence to support this. Art Arnold down at UCLA, many of you know, have created this mouse with the SRY, where he's able to separate both the genetic sex and then also the testosterone influence here. And he found also in the animals that specifically from sex chromosome complement, or sex chromosome influence, you can have differences in a number of behavioral phenotypes, whether it's aggression, pain perception, things like autoimmune or neural tube defects as well. So if you look at the genetics, it's really, there's a good framework to really thinking about why this might happen. And you know, the X chromosome is substantially larger than the Y chromosome. So the estimation is that it's about 2,000 genes compared to 200 genes on the Y chromosome. And probably why it's even possible that we have monosomy and why there's more of a balance. You know, you would think that because of the size of the X and Y that males would be hugely disadvantaged. But evolutionarily, that's been compensated for, right? We turn off one of the second X chromosomes. So there's this inactivation effect. So what might be driving this in Turner syndrome? They still have one X chromosome. Typically developing women, one of those are turned off, but yet there's these huge differences in phenotype, right? And even in Klinefelter syndrome where that second X is turned off. Why are we seeing this? And the theory has been that it's really because of these regions, the pseudo-autosomal regions in the X's and the Y's, and in these X escapee genes that don't get turned off in the second X chromosome, that might drive this gene dosage effect. And this has been illustrated beautifully by this one study by Rao and Dr. Appold back in '97. This was the SHOX gene. So the premise behind the SHOX gene, which is in this pseudo-autosomal region, this region that doesn't get inactivated in the second X chromosome at the very tip, you can see it moderates stature. So having one X chromosome, Turner syndrome, a classic part of the phenotype is short stature, and then in XXY, where you have 3 copies of the SHOX gene, it's overexpressed, they tend to have much greater stature than their typically developing males. So this is a nice example of how a single gene, so stature is obviously mediated by several genes, but this is one example of how you can see this effect where it would be enacted. What's really disappointing is since the SHOX gene was identified in '97, there's been almost no follow-up to finding any single gene candidates really to explain something else that would be kind of identified across all 3, Turner syndrome, Klinefelter, and typical development. So, you know, there are certain genes that we know are implicated in a number of disorders, color blindness, Fragile X syndrome, hemophilia, and on the Y chromosome, it's more related to sexual function, so infertility and gonadal dysgenesis. But the X chromosome, we know there's so many genes out of those 2,000. It's super enriched for genes related to cognition. So we know probably there's somewhere between 800 genes or so that are related to intellectual disability, and 200 or 300 of those reside on the X chromosome, right? So it has an outsized effect on cognitive development. And it makes it a very compelling locus for us to try to understand better why there might be some of these sex differences, particularly in the PAR regions. But when you look at the sex chromosome literature, people have really tried to isolate whether there's any single genes associated with it, and it's been disappointing. It hasn't been replicated. So whether it's just the phenotype associated with Turner syndrome, fear recognition, these social cognitive traits, it hasn't been replicated in Turner syndrome, and only very weak evidence, very small sample sizes in Klinefelter syndrome. So along with our collaborators in genetics, Alex Urban and his postdoc Zhang Long, we took a bunch of the kids that we put in through our neuroimaging sample. We took a subsample of them, and we ran RNA-seq on the samples. And just for a nice proof of concept, we're looking at the X chromosome expression. You can see the exist is much decreased in the XO, the Turner syndrome girls complain to XX. And this is great proof of concept. Exist is the gene that determines inactivation of the sex chromosome. So the fact that they only have one X chromosome, there's way less exist expression. Perfect. Same thing in Klinefelter syndrome. You're looking at much increased exist compared to the typically developing males because they're turning off that second X chromosome. Great. So we looked and we really were hoping there's going to be a smoking gun here. We're going to find something in the pseudo-autosomal regions that's going to nicely tie everything together. And while in fact we did find that the same genes that are downregulated in XO are the same ones often that are upregulating XSY in the PAR, none of those genes are really related to cognitive function. But you know, that's not a big surprise because we're looking at peripheral red blood cells and not neurons. So there's really not too much weight we could put into that. But it gives us a premise to think that there might be really this dosage effect if you study neurons, and that's an area that we're looking into more deeply. But here's what's interesting. If you look across the entire transcriptome, there's a huge number of sex differences. So everything across the red line, these are differences in expression across known annotated genes, right? And there's approximately 304 that are different between Turner girls and typically developing females, and about 97 that are differentially expressed between Klinefelter boys and typically developing boys. And it's occurring not as we had originally hoped or hypothesized just on the sex chromosomes, but across the entire genome, transcriptome. So what we did is we did this comparison. We did this basically a conjunction analysis where we looked at with the idea that because they have a different number of X chromosomes, so Turner syndrome girls relative to typically developing girls, X to XX, Klinefelter boys to XY, XY to XXY, and males and females, XX to XY. In that same relationship, if you look at the differentially expressed genes, their RNA-seq profiles, maybe we could find some kind of pattern that distinguishes them, where there might be a convergence on certain genes. So, if you look at all the differences in the genes, that ones that similarly were differentially expressed across all three came down to these 31 genes. And remarkably, we found exactly what we had anticipated, which is all of these occur in the direction that we had thought, right? So this red line is the difference between XXY and XY. So the average fold change in the expression is much higher in the XXY compared to XY. And in those same genes, XY versus XX or X versus XX, it's happening in the direction. It's underexpressed, those same genes. So, this is, I think, these are only a small, I think only about 2 of these genes are actually on the X chromosome. Across the entire transcriptome, there's a variety, there's at least 31 genes that are converging in terms of the pattern that you would expect, despite the fact that there's no haploinsufficiency or an abnormal number on those other chromosomes. It's all driven, presumably, putatively, by the fact that there's a difference in the number of X chromosomes here. So one of the things that, we're looking into now is what are the network effects that's driving this pattern? So just to wrap up, I know I'm out of time. Hopefully what I presented a little bit today is to get us thinking a little bit about both domains in terms of phenotype, so behaviors that aren't sex type necessarily, and also mechanisms that aren't related directly to hormones, but more directly to sex chromosome genetics. That might make us think a little bit about some of, this will fill out the universe of ways we think about sex differences, is that there might be other ways, downstream effects that derive from the sex chromosome genes that are, in network or gene-gene interactions affecting genes across the entire genome, whether it's through epigenetic effects, methylation, whether it comes back and converges on other hormonal pathways or other transcription factors that also contribute to these sex differences. And again, I just wanted to thank Alan, who's the director of our center, all the postdocs and RAs, my collaborators in genetics and psychiatry, Joachim, Alex, Zhang Long, and endocrinology, Tandy. Thank you very much.